Okay, so where we left off last time was the end of chapter 5. So we're going to be getting on to chapter 6, talking a little bit about chromosomes, but first some housekeeping details. I have not put up the homework for, or put up the grades for September 19th for the homework yet, and I will do that here hopefully tonight. I have it all in a Excel document, but I just haven't put it up yet. So I will grade that. If any of you were wondering why you had zeros, it's because I hadn't put the grades in yet. I'm going to put up 2 homework assignments tonight for you to do, and I will put them on Springboard under quizzes. And they will be due by Sunday evening at 11:30. So I will tell you, you can pick one. If you pick both of them, that will give you extra points. If you missed any of the homeworks so far, then that gives you an opportunity to make up those points. So there's going to be one set of homework for Chapter 5 and one for Chapter 6. So you can choose to do one of them. You can choose to do both of them. That'll get you guys all caught up on your points if you missed one of the homeworks. So I will put it, like I said, it's on Springboard, and it's under quizzes. And it will be due by Sunday at 11:30 p.m. So hopefully that will give you 11:30 p.m. Yes. 11:30 at night on Sunday. Because hopefully everybody will be in church at 11:30 in the a.m. on Sunday, or wherever you would like to be. Some of you will have wonderful sleep to attend to. Some of you will be playing outside, raking leaves. Whatever makes you happy on Sunday at 11:30 a.m., it does not have to be your homework. So I didn't end up putting up the lecture from last time. I actually spent all day Wednesday in the emergency room. So I've talked a little bit about my my health issues, but so I'm diabetic and I have problems. I've actually had 5 abdominal surgeries, and because of that, when they start cutting into your abdomen, you get your things growing back together. It's called adhesions, and so I have adhesions in my abdomen, and they make my bowels move around, and so my bowels did something crazy Wednesday night, woke up at 2 a.m. and said, I think I need to go to the ER. So I spent all day in the ER on Wednesday, and they sent me home with medicine. So I guess if I seem really happy, I'm really happy. So yay me. But there goes that, what were we talking about? The endoplasmic reticulum in the liver making you become kind of, so that the medicines don't work anymore. I start to have that problem. I have a couple of student emails that I haven't answered yet. So if you sent me an email yesterday, I had like 150 student emails. So I apologize if I didn't get to your emails yet. So I will get to those hopefully tonight. And then I also have to input the interim reports. So I will be putting up grades. When I finalize the grades, I'll send you guys an email, and I'll put up those interim reports. Basically, the university wants to know that you're either doing well or you're not doing well. They want to know for athletes. They want to know for students in particular programs. And so I do interim reports for everybody, not just for any one segment. So I'll put those up, and I'll definitely be available to answer any questions, or if you need office hours, or if you just need to ask about it via email, I will definitely get to that. So that's what's on my to-do list. Is there anything else you guys need me to add to my to-do list? Yes? Do you know when the next study guide will be available? The next time we have a test is October 9th, and so I will try to get the study guide up at least a week beforehand. So if you want to send me an email to poke at me to do it, then I tend to remember things in my email. So I was hoping to have it the weekend before, because I think last time you gave it to me a couple days before. Yeah. Send me an email, and I'll get it up there as soon as possible. Thank you. Yes. I will try to, yeah, that's definitely better to have it the weekend before. I agree. Okay, that's a good idea. Anybody else have good ideas or ideas? Okay. So moving on to Chapter 6, we're going to talk a little bit about chromosomes and cell division. And for those of you that have already had your lab, this may seem like a review of the things that we've been talking about in lab. But this is definitely, since you're going to hear it twice, it's definitely that's something important for you to do. So we're going to be talking about how traits are passed on from parents to offspring. And so we're going to be talking about two things that I think students are often confused about because the words sound so similar. We're going to be talking about mitosis and meiosis. And mitosis is basically one cell splitting into two cells, making an identical copy of itself. And then meiosis is how we make the sperm and the egg. And I will try to make those as abundantly clear and different to you as possible. So we're going to talk about this a lot in terms of cancer, because cancer is one of the most defining diseases that have to do with uncontrolled cellular growth. You may have thought about your cells in terms of why do cells die, or how could we make cells live forever? How can I be as healthy as possible? And we're going to talk first of all about immortal cells. What would happen if your cells could actually live forever? And so part of the reason that your cells do not live forever is because they have something inside of them called telomeres. And basically you've got these long strands of DNA, and they have caps on the end of them. And the cap on the end of the DNA, like you see in the electron microscope picture right up here, the orange spots at the end of the chromosomes are the telomeres. And the telomere is this cap on the end, which says to the DNA, no more dividing, no more making more of yourself. And it's this buffer zone where every time the cell divides, it gets shorter. So in human cells, that means that our DNA can divide about 50 times. And after 50 times, so much of this telomere breaks off. Every single time the cell divides, a little bit breaks off, and then another little chunk breaks off, and then another little chunk breaks off. And when eventually we get to the point where no more chunks of the telomeres can break off, that's when we get cell death, because we can't have any more replication of that DNA. So much of that DNA is lost at that point that we can't make another copy of it. And what we consider it as is a protective cap at the tip of every chromosome to tell our cells, stop making more of yourself, because at a certain point there would be too many mutations, and your cells wouldn't be providing accurate copies of themselves. So every time a cell divides, a little chunk of this end cap breaks off until finally, if you don't have any more cap on the end, then the DNA breaks, and it doesn't pass along enough information to give a fully functioning copy of that DNA. So what a telomere is basically is a non-coding repetitive piece of DNA that serves as a cap. So each one of these little X's that you see up here has the little golden color. It's the cap called the telomere. Makes it so that once it breaks off, then the DNA doesn't replicate anymore. So there's a disease that is very interesting. If you've ever heard of this disease called progeria, progeria is a disease where children age very rapidly. In this case, this is something like an 8-year-old girl, and she has basically the cells of an 80-year-old. So she aged very, very rapidly. What? I think that that's it. There was somebody that Howard Stern used to do interviews with, and I think that's the movie you were talking about with Brad Pitt, right? Yeah. Didn't he go in reverse? Like he started off really old and then got really young. This is where they age so quickly, like a 2-year-old is like a 60-year-old, and then they get, they can rarely live to be past 13 because their telomeres are so shortened that it's like their cells age very rapidly, and they get all the same diseases that people get when they're in their final years of life and rarely live to be past 13. And so it's such a sad disease because she looks like a very old person, but she's, she's still very, very, very young. And there's lots of interesting information out there about why that happens, and I just think it's something that's so sad. It's similar to what you would consider cancer cells to be. Cancer cells are considered to be cells which have forgotten how to die. So basically that keep replicating even when they should be shut off, even when they should be done, and then cause tumors, and tumors which can suck so much energy and so much resources from the cells that they then take over the body and can cause death in the individual. First thing that we need to differentiate between, or look at the differences between, are chromosomes that are circular and chromosomes that are linear. If we are looking at chromosomes that are circular, those will be our chromosomes that are inside of bacteria. So bacteria have that circular chromosome right in the middle that's just kind of wound up, and they have no organelles, just one simple cell with a round piece of chromosome in them. And then in animal cells, plant cells, their chromosomes are linear, which means that they look like those X's that we usually think of when we think of our chromosomes, and what is the backdrop for this PowerPoint presentation. And so here's the two different cells. In the prokaryotic cells, the primitive cells, the bacteria cells, that's where we have our singular circular chromosome that's attached to the cell membrane. So it's just a circle inside of the cell, and it's stuck in certain spots to the cell membrane that's around the outside of the single bacterial cell. And then we have our eukaryotic cells, our plants, our animals, some of our protists, our fungi. And that's where we've got our chromosomes in different numbers that are found inside of the nucleus of the cells. And they are in those distinguished X's. And they've got what's called histones. And those are the end parts of the pieces of DNA that are the chromosomes. When they're inside of the cell, they just kind of look like linear pieces of DNA hanging out inside of the cell. It's not until they actually get ready to separate into two individual copies that they begin to line up like those X's that we think of. So prokaryotic cells, when they're making more of themselves, are dividing by what's called binary fission. So basically, binary, you see the word bi and you think two. So it's fission, fizzing down the middle, making a fissure down the middle, and separating into two individual cells. So the way that bacteria reproduce is basically splitting down the middle and making two exact copies of those cells. And so here's a picture of what that looks like. Here we have our actual electron microscope picture of it. And here we have what we would consider the parent cell. So in the case of bacteria, we don't have two parents. We only have one parent. Our prokaryotic parent cell goes about doubling its DNA, and then it's going to go through replication. So this is an interesting process, and it's kind of hard to explain what it is that you're looking at. So here you've got your piece of DNA, and it's in a circle. And what basically happens is one side of the circle pulls apart from the other circle. So you can see how if we had two strands of circular DNA that were attached together, and you were to pull it apart, it would unwind. The new pieces of DNA would come in here, and they would form two separate single circular pieces of chromosomes. and then those two pieces would be there in the middle of the parent the parent prokaryote, the parent bacteria. The cell would begin to elongate, so the bacteria becomes longer. He pinches off. Like I'm assigning that he's got a gender. So the bacteria pinches off here in the middle. One of the circular pieces of DNA goes in this direction. The other goes in this direction. This is called cleavage. So if you think about what you think about when you think about cleavage, you've got your two pieces of cleaved DNA down the middle, and what's called the daughter cells are formed. So we go from having one long parent, cleaves down the middle, and then we've got two separate bacterial cells that are exactly identical to one another. And we do that through the process called binary fission, taking one parent, binary, going to two cells, and they are genetically identical. So cell division has to be preceded by replication. You've got your normal number of chromosomes inside of those cells, and you have to make another copy of them before you're going to split into those two cells. So in order to persist or keep along the species line and propagate, make more of yourself, you have to make more of that DNA first. So the process of replication is the process of DNA duplication. And I try to explain these words a couple of times because I know that this is like a vocabulary test here to hear all these words, especially if you're not familiar with them. So when you think about replicating something, you're making more of it. So making two exact duplicates, two copies of this cell. So they have what's called complementarity. So when something complements something, when you have one color complementing another, it means that the color goes with it. We've talked a lot in lab and a little bit in here about complementary bases. So the characteristic that makes DNA double-stranded is that one side of the DNA goes with the other side. So they are complementary strands. Every time you have an A, it always goes with a T. Every time you have a G, it always goes with the C. So that's your Austin, Texas is a great city, for those of you that have heard me say that before. A goes with T, adenine goes with thymine, and then G goes with C. Guanine goes with cytosine. So where you have one strand, you can always predict what's going to be on the other strand because those things always go together. They are complementary, and that's called complementarity. Big words. So we've talked about DNA replication a little bit when we talked about cells. We have our double-stranded piece of DNA that's in this double helix. So we have our chains to our DNA, and the first thing that happens when they're getting ready to duplicate is that they have to unwind. So something comes along just like a zipper and unzips the DNA down the middle. So unwinds it and separates it into two strands. Then these nucleic acid bases are floating around here in the cytoplasm, and they come in and the A's match up with the T's and the G's match up with the C's, and they basically go about rebuilding themselves. So there's an enzyme that goes through and unzips it and pulls it apart. Then the bases start attaching, and it rebuilds itself into two identical strands of DNA with these nucleotides that are floating around in the cytoplasm. So that's how we grow new strands of DNA. And this purple spot right here represents an enzyme. So we unzip it, and then we rezip it on each side. Then we have our single strand right here has two exactly duplicated matching strands that are identical to the cell's original that can go into each of the new cells. So as we've talked about with mutations, errors sometimes occur when the DNA is duplicating itself. As you can imagine, they're billions of strands long, and so there's always going to be some errors. We find that DNA a lot of times can go through and fix itself. It's got enzymes that go about fixing it. but sometimes we have really bad mutations, like we talked about with the point mutations, and we talked about some of the diseases that can occur. But as we have also talked about, this can be a good thing when the mutation leads to an adaptation that makes it better for the thing to be able to survive. So, we have these variety of errors that can occur during replication, and these DNA processes that go about repairing the DNA. But if the error remains in the sequence and it's replicated or made over and over and over again, that can make the individuals different than their parents. So, so this DNA that's found inside the parent, if you get this mutation in it, that can make the offspring different than the parents. So most cells are not immortal. This process of mitosis generates replacements. So if you were to go around this room, and when they sweep these rooms at the end of the night, you see these big piles of dust, and you may think, well, where did that come from? It's not necessarily the dirt that we all track in on our shoes. But every single one of us that's in here right now, our skin cells are going through mitosis. They're making more of themselves because we're repairing. If you have cuts in your skin, or you have places where your skin has worn down, and we're always growing and developing. So the dust that we see all around us is the skin cells of ourselves and our friends. And so you are actually the person that is making the dust in this room. And it's kind of freaky to think about that we have our cells that are just being sloughed off all the time. But just by scratching your skin, you can take off thousands of skin cells, and it will head out into the environment all around us and make everything dirty. So isn't that exciting? Mitosis really just has this one purpose. To be able to generate these new genetically identical cells. If you think about it, your skin cells, when they're making more of themselves, you want them to make genetically identical cells because you want your skin to grow on you nice and fresh and healthy. And we do that so that we can grow, and we do it lots when we're young. We do it when we get older as we're replacing our damaged skin cells. We're being damaged all the time by bumping up against things, by being out in the UV, or the UV radiation. We are continually growing and continually replacing, and that's why if you think about what like ladies' skin lotions do, as you start to get older, your skin starts to become thinner because the cells aren't replacing themselves as fast. And so the skin lotions that are supposed to plump up the skin and make it look nice and fresh and new, or if you've ever seen somebody that gets a facelift and they get those chemical peels where they burn off the outer layer of the skin and the new cells come in from underneath and go to replace those old skin cells. And so we're constantly having mitosis going on so we can replace the skin cells on our body, replace our red blood cells, replace all the cells that are inside of our body. And if we stop replacing the ones that are damaged, then that's where we start to go through aging or start to go through, run into trouble. So we are constantly in a need for new cells. If you think about plants, as they grow up bigger and bigger, they're going through mitosis, replacing those plant cells. If you think about the roots of plants, if you've ever seen how they weave their way through the soil, right there at the tips of the roots is where mitosis is happening the fastest, as they're moving their way through the dirt and reaching out to get more moisture. If you shave your face, shave your legs, the act of shaving is basically damaging those skin cells that are right around your hair follicles. As you shave them off, you're basically ripping off your skin cells. And so those cells have to be replaced. Otherwise, you're going to run into trouble. You can get infections. You can have all kinds of problems. but your skin cells have to be constantly replaced, and just the act of shaving is damaging them, so you have to replace them. The process called apoptosis is where your cells all have a certain age at which they are planned to die, or we call this cell suicide. So your cells are going to commit suicide after about splitting apart 50 times, and certain cells within your body are targeted for apoptosis, and they happen in a certain progression as you go through your body, and we're going to talk about that here. But apoptosis is cell suicide, or cells pre-planning to die off. So in this process of mitosis, where we've got our cells that are being replaced by mitosis every day, we've got our red blood cells, and those are being constantly replaced. And that's very important to you being able to survive and carry the oxygen around in your red blood cells. And the rate at which this mitosis occurs can vary dramatically depending on where it is. Like inside of your bone marrow, that's where you're creating most of your red blood cells. Inside of the bone marrow is where mitosis is happening very quickly. On your skin cells, that's where mitosis is happening very, very quickly. As you're a child and you're growing and developing, making brain cells really rapidly. That's one of the reasons they say that drinking is really harmful when you're young, because if you damage those brain cells that are growing so quickly, that can have effects on the rest of your life. So children who do drugs at a very young age, or who are exposed to drugs, that's often very detrimental to them, because that's when their cells are dividing the most. So mitosis leads to these duplicate cells, and this is an example of cells duplicating. We go from one parent to two daughter cells. And so that should make sense to you. We go from one cell to two cells as they cleave and pull apart. So this is the process, and I know it's very small, it's kind of hard to see, but we have our parent cell that has all of the chromosomes inside of the nucleus. During what's called interphase, the genetic material is replicated, so we go from this set of chromosomes inside of the nucleus. We are starting to make more of those chromosomes. Then, as the linear chromosomes line up in the X's right here, they're going to begin to line up along what's called the equator of the cell. Just like we have the equator of the Earth that's right through the middle of the Earth, separating the two hemispheres, all the chromosomes line up here along the equator of the cell, and we start to have what's called spindle fibers. These fibers that are attached to the poles of the cell, and the spindle fibers are going to pull on each side of the chromosomes until at the third point right here, you can see half of the chromosomes go one way, half of the chromosomes go the other way, and they pull the duplicates apart. Then finally, since we have half the chromosomes going one direction, half the chromosomes going the other direction, the cell starts to cleave apart there in the middle until eventually we have the two nuclei that are forming around the genetic material, and we have a complete and exact copy, basically twins, of the new cells that are having the exact same genetic material inside of them. So mitosis is this four-step process, which is what I just showed you. So here it is a little bit more in detail. In the preparation for mitosis, that's where our replicated chromosomes are there inside of the nucleus. We've got our nuclear membrane that's all around those chromosomes in the parent cell. And here is where we see these two little things here are called the centromeres. And these are the poles of the cell. If you're thinking about it like the Earth, one is going to go one direction to the North Pole, and the other is going to go the other direction to the South Pole. And the chromosomes that are here in the middle are getting ready to separate into these two exact copies. And so they are going to be pulled apart at this junction of the X, right in the middle. And they are going to go from being one single chromosome to two copies that are attached right in the middle. And they look like this X because they are attached at the middle at the centromere. And what we call these, the specialized vocabulary for it, is sister chromatids. The centromere is where they're attached. We've got our two linear pieces. What? Oh, I'm sorry. Bless you. I thought you said what? So we've got our centromere right there in the middle, and all of the genetic material has an exact copy. Think of them like identical sisters. They are, well, I guess it would be identical sisters that are attached. They are attached right there in the middle. They are identical replicated copies, and they're joined together in the middle, and they're being prepared to be separated apart, being prepared to be pulled from one side to the other side. And so this process called interphase, that is where everything has replicated and it's ready to begin this process of being pulled apart. We go then through mitosis. The nuclear membrane has to break down, so the DNA has to get out of the middle of the nucleus. So the nuclear membrane breaks apart. We've got our spindle fibers. We've got our spindle fibers here that are each attached to the centriole. And this centriole is going to the North Pole. This centriole is going to the South Pole. The X's are all lining up here along the equator, and they're getting ready for half of them to go one direction and half of them to go the other direction. So then the nuclear membrane is completely dissolved. Everything gets lined up here right before metaphase in prophase, so this period where the nuclear membrane's breaking apart. Everything's in its X's and the poles are going the two directions. Then we go through metaphase, and metaphase is where we've got our equator. We've got our North Pole and our South Pole, and the chromosomes are lined up and getting ready to be ripped apart. And the next, right after metaphase, is when we go into anaphase. Anaphase is where they have actually been ripped apart and they're getting pulled towards the poles. We've got one full set of chromosomes that are going one direction and the other full set of chromosomes going towards the other direction. Then we go into telophase, where the cell begins to actually split into two. And we've got half of our chromosomes on one side, the other half of the chromosomes on the other side. The nuclear membrane begins to form around those chromosomes. So where there had been no nuclear membrane before during anaphase, we've got the nuclear membrane that's forming again during telophase, and the cell is beginning to pinch in two during cleavage formation. And then finally, we get our complete pinching off, and the two daughter cells are going to go their two separate ways. And cytokinesis, as you can imagine, when you go from one big parent cell and you split into two, you're going to have all the parent cell is going to become half of the size. So the cytoplasm is going to begin to make more of itself to make two full-size cells, and so that's basically where the cell is building itself so that it can be equal to the parent cell. So when we have this process happening out of control, that's where we get things like cancer coming into play. So cancer is unrestrained cell growth and division. If you think about what you know of cancer, when people find out they have cancer, they usually find some kind of tumor. In breast cancer, women find lumps. Well, men can also get breast cancer. When you have these tumors, you have these abnormal formations of cell growth and division where they're sucking the resources out of the individual. And it is the second leading cause of death in the United States. I think it's 20% of all deaths in the United States happen because of cancer. And the leading killer of people in the United States is heart disease, which is hereditary, passed on from parents to offspring. So cancer is on the rise, but ways to treat cancer is also on the rise. This is what it looks like when a cell is going through cancer. If you were to take and grow normal cells in a petri dish, they would keep dividing until they bounce up next to one another. And at that point that they reach the other cells, the cells begin communicating with one another, and they say, hey, we're right next to each other, stop dividing. And so they just make a thin, even layer, and when cells come into contact with one another, they stop dividing. Cancer cells, on the other hand, when they come into contact with one another, they don't recognize that they're right next to another cell. And so they ignore this signal that they're right next to another cell, and they continue to divide, basically piling up onto one another. And that's what makes for a tumor. So here, if we've got our normal cells, one parent cell becomes two daughter cells. Then as each of these become parents, they divide into 2, and they divide into 4, divide into 8, until eventually the normal cells are no longer able to divide, and they have this like a normal pattern of growth, and they stop growing. That doesn't happen in cancer cells. As the cancer cells grow and divide, they don't do so at a normal rate because they don't recognize each other as other similar cells. So they continue to divide indefinitely. They can divide forever and ever and ever until they overwhelm the host. That's why people that get cancer often get so weak at the later stages in their life, because tumor cells like this are just going to suck the energy and suck the resources out of the organism. And they are going to continue growing out of control, making these tumors bigger and bigger and bigger. And it says here the membranes of cancer cells tend to have reduced adhesiveness. Adhesiveness is this process of being sticky. So they stick to each other less than do non-cancerous cells. So whereas normal cells make these nice little patterns, these cancer cells just keep growing out of control and make these big tumors, and they will grow forever until they overwhelm the host. So piling on top of one another. So cancer cells have several features that distinguish them from the normal cells, which is what we just kind of went through. And there is a difference between what's called a benign and a malignant tumor. Metastasis is the process through which cancer cells, which are the cells that they show here within the body in white, and so metastasis is where they separate from a tumor and move throughout the body. If you've ever heard of anybody that's had cancer in their lymph nodes, that tends to be one of the cancers that spreads very easily to other cells around the body. Because as it gets into the circulatory system, it moves and circulates throughout the body, and that's called tumor metastasizing. That's where it spreads around the body, and it says prognosis worsens. Prognosis is like the chance of death. If something is a benign tumor, that means it isn't going to spread throughout the body and take over and overwhelm the body. But a malignant tumor, a cancerous tumor, that's the dangerous kind of cancerous tumors. And so metastasizing is when it spreads through the body in the circulatory system.
So basically what you're saying is all tumors are essentially cancerous in the fact that they come from cells that are still dividing.
Right, but it's not cancerous until it's sucking so much of the resources from the body and growing out of control. So you could have tumors that are benign, that are non-cancerous, and they are due to the same process, basically the same process, but if they're benign, they're not going to spread throughout the body, and they're not, it's not something that you could kill off with like chemotherapy or radiation.
So why is it that benign tumors don't spread? What makes them
I don't know that I could do justice to explaining that. I will look up more for that, and I'll see if I can explain it better the next time. I don't want to give you a bullshit answer, but I could probably make something up that would sound good. I don't want to do that to you. So I don't know, but I will find out. So cancer, this uncontrolled growth and lack of cells recognizing one another. So here we've got our danger of cancer cells, where we have, like in the example of the lungs, if we have a malignant tumor developing in lung tissue, as it gets larger and larger, that space that you need for your lungs, that you need to extract all of that oxygen that your body needs, that tumor isn't doing what the lung cells are doing. And you need those lung cells in order to be healthy. And so as that tumor takes up more and more area, you're getting less and less oxygen that you need, and it's taking more and more resources from your body. And so it basically impinges on your other cells. It can disrupt the functions, like the extraction of oxygen that you need in order to be able to survive. And eventually, if it takes up too much room and it doesn't allow you to breathe, then that's going to be disastrous. So one of the awful things about cancer, and from anybody that's ever known anyone that has cancer, cancer is bad, but the treatment for cancer is just as bad. Treating somebody that has cancer, for some people who are healthy before they get cancer, it can be not as harsh, but for children or for people with weakened immune systems or for people that are elderly, when you go through radiation and when you go through chemotherapy, it can be just as devastating to the body. Introducing through chemotherapy chemicals in the body that are going to kill off the cancer cells, it's going to do a lot of damage to the body, to the regular body cells, as well as the cancer cells. And so that chemotherapy, it's going throughout your entire body. And then radiation has definitely made cancer more treatable, because when you radiate the cells, you can often do so right at the specific point where the cancer is. So radiating skin cells is easy. If you have a malignant skin cancer, then treating it with radiation, I don't want to say it's easy, but it's easier to do than if you had liver cancer or lung cancer. If you're trying to radiate the lungs, they are inside of your body and they're hard to get to. So that's why lung cancer is such a killer. Liver cancer is such a killer. Pancreatic cancer. Whenever they get to the internal organs, they're often very, very hard to treat. If they're in your bone marrow, that's one of the most difficult. And unfortunately, that's one of the ones that kids get so often. And it's so awful. I don't know if I ever told you guys this. My brother is the donor relations coordinator for Akron Children's Hospital. So he goes out and collects money to help Akron Children's Hospital grow. And he works a lot with these kids. We go around doing fundraisers and trying to raise money for them so that no matter who goes into the hospital, even if they can't pay, Akron Children's Hospital won't turn anyone away. And some of these stories about these kids with cancer is just, it's unbelievable. It's amazing. Cancer, unrestrained cell growth and division, they are growing, not recognizing each other, and dividing out of control and forever. They can lead to these large masses of cells called malignant tumors, and those cause all kinds of health problems. And then the treatment to focus on killing them or slowing them down through chemotherapy or radiation is often just as devastating as the actual disease. So that's where we're going to end up there, I guess on a high note. That's kind of a depressing place to end. But if there's anything else that we need to talk about, or if you want to come see me and discuss anything, then I'll be around here for a little bit. And if you're not reading, if you haven't read through chapters 5 and 6, you should do that. Do it. What? Well, thank you. I didn't write the textbook. I wrote the